this little thing in the way. It's kind of in the way. Here we go. So, Justin. All right, thank you everyone for sticking around for my presentation. Before I get started, I'd like to thank Karen Mock, who helped me take on this little project, and my PhD advisor at the time, James Long, at Utah State University. I'm going to be talking to you today about the potential implications of radial growth differences in different ploidy levels in the Pando stand. Now you've probably heard, you're quite familiar with. There's a picture of the Pando stand. We're all aware of the diverse, heterogeneous environments that Aspen create, especially in western landscapes. They tend to be hotspots of diversity. They also, as we've heard earlier from Bob Campbell, they're also great areas for forage production, wildlife habitat, and they're thought to be particularly resilient to large-scale disturbances such as fire or coppice harvesting, and that's primarily a result, we think, of their ability to coppice or to sprout vegetatively. However, there's been some recent relatively recent events, sudden aspen decline, other global change type drought events that we think might be resulting in some decline. Other factors such as succession, insects, bugs, and crud might be affecting these stands, so it's causing us to rethink our approach to aspen management. And as you saw in Karen's presentation, there are other reasons to rethink aspen management. This is a thoughtful endeavor, especially based on this recent genetic work which essentially indicates that aspen stands, as we see them now in the landscape, are the result not just of vegetative reproduction, but almost certainly some sexual reproduction. And what we find in these large stands is that triploids and diploids are present on the landscape, and that makes us think that's cause for concern about whether we're promoting sexual or asexual clones on the landscape. There's also some indication from the literature that there might be some growth differences between these types of clones. And so that's where my work comes in. We're going to dive right into the Pando stand and look in detail at the individual ramets from different clones. And so where phenotypic differences were traditionally used to differentiate between clones, we've capitalized on the genetic work that Karen Mock's lab has done, and we're going to make comparisons between aspen ramets that we know to be diploid or triploid. And specifically what I'm going to test is within this Pando stand whether or not there are differences in those stem ages based on the radial growth and radial increment counts, whether there are differences in the environment where those stems are located, whether there's difference in growth over time, and you'll see why that's necessary in a minute, and then whether this response to growth has anything to do with climate, and by climate I'm talking about growing conditions, growing season, precipitation, weather, things like that. So to conduct this study, what we did is we went back to Karen's 50-meter grid and we revisited these stems, and we built two populations based on her genetic data, triploids and diploids. We relocated the stems, which were marked conveniently. We determined whether they were male or female, with some help from Bob Campbell telling us when to get out in the field. We did some basic metrics of the local environment around those stems, and we measured their size, and most importantly, from my viewpoint, was taking the increment cores of these from these stems, and this was done using traditional dendrochronological approach, where we in our case sampled one increment core per tree at roughly 1 meter in height, and then those cores were taken back to the lab to be processed. And this is essentially how those cores were taken in the field, for any of you who may have never seen this before. Voila, data. In the lab, the increment cores were processed using standard techniques, sanded with progressively finer sandpaper, right under digitized, read and digitized under a microscope. All the cores were cross-dated for temporal control of ring width increment over time, and standardized using some pretty approaches. And then for our analysis, I tested for differences of ages, which were determined from the increment cores between the diploid and the triploid samples. We tested for differences in diameter, differences in the local environment, which I'm calling relative density. We tend to think of stems per hectare, but also, or excuse me, absolute density, stems per hectare, but also relative density, how well stocked the stands are. And then using the annual growth data, we looked at differences between the diploid, potential differences between the diploids and the triploids. And then whether there was an effect of climate, things such as specifically monthly precipitation, monthly temperature, and monthly drought stress on the growth over time between those populations. So getting into some results, to start, stem ages between the populations did not differ. This suggests that most of the stems regenerated relatively close in time after some large-scale event in approximately 1870, perhaps a fire. The current relative density in these stands also did not differ. They were both fully stocked, full site occupancy. So there was competition-related mortality in both stands, both the diploids and the triploids. However, there were many more stems per hectare, absolute density in the diploid versus the triploid. And the concomitant result of that is that the mean stem size in the triploid was much larger than the diploids. So we're thinking the diploids might be growing faster. This slide in and of itself has pretty strong implications for how we think about aspen growth on the landscape. But some more results. If we take the ring increment data and standardize it since the first year of growth, which seems appropriate since they established during a relatively short window, we see that the triploids had a strong growth advantage, especially early in stand development, over the triploids. And I did some quick numbers here. During the first 50 years, 86% of those years were statistically different, higher growth for the triploid. In the first 100 years, 70%. And the effect seems to attenuate over time as aspen ages, like you might expect. Another way to look at this data is just to bin it up into groups based on individual ring widths. And what we see is that the triploids, which are in black, by and large had substantially more annual increment than the diploids. And again, this is manifest over time, but it attenuates over time. So moving on then, is the rate of growth affected by climate? We addressed this by using what's called correlation function analysis. And what you're looking at here are correlation coefficients between monthly, in this case, monthly precipitation data and the standardized ring width index for the diploids and the triploids. And we're looking at the current growing season in the right side of the panel and the previous growing season in the left side of the panel, since there's a lot of temporal autocorrelation in tree growth. And we can see that there doesn't appear to be a very strong difference in the response to precipitation between diploid and triploid aspen. And I'm going to argue that's the same case can be made for the response of both diploids and triploids to drought, Palmer Drought Severity Index. Now there does seem to be a little bit of a difference in growth as a result of monthly maximum temperature, and as you might expect, growth is reduced when temperature goes up. That's why we have a negative correlation here. And for the diploids, that occurred during many of the growing season months, whereas not so much for the triploids. So what does this mean? What's happening in the Pando stand? My interpretation is that there was a large-scale event, probably a fire, in the 1870s, which resulted in the establishment of the Pando stand. Probably the triploid and the diploid individuals represent both sprouting and sexual reproduction, seedling events in that stand. Over time, the triploids grew faster, attaining larger individual tree sizes, but lower absolute densities than the diploids. This begs the question, what are those differences? And in this exploratory work, I'm not sure we've nailed down what that could be, so I'm going to offer to you some alternative hypotheses why we see those huge differences. The first is that perhaps the diploids are more sensitive to those summer temperatures, and that cumulatively over time, this results in a reduction in their growth. While this is probably true based on the data, it fails to explain that marked difference early in stand development of growth between the triploids and the diploids. So a second alternative hypothesis I'd like to propose is that there are inherent genotypic differences. Now I should mention that virtually all of the genotypes that we sexed were male, which we expect at this elevation. So that's probably not driving the difference. However, these data showed that among our triploid samples there were 2 genotypes, and among the diploid there were 14 different genotypes. So it appears there may be a trade-off here between genetic diversity in this particular landscape and the growth capability. A third hypothesis I'd like to propose is that the triploids, if we assume that there was both vegetative reproduction and seedling reproduction, that perhaps the triploids had an inherent advantage from the outset because they're coming from previously established root systems. Whereas the seedlings, in addition to fighting for light, would also be developing their root system, and perhaps that explains the growth differences over time between those stands. This might also have some implications for clonal rate of spread if the triploids grow faster, but our data don't address that directly. So, in conclusion, what I would like to include with is a statement that in the past, aspen diversity is likely a result of post-disturbance regeneration ecology that included both sexual and asexual reproduction. And in the continued presence of disturbance, we're likely to see that type of pattern. In the absence of disturbance, it's hard to say whether we're going to, what kind of reproduction strategy aspen might rely on. But I'd like to make the suggestion that if we're going to confer an evolutionary advantage to aspen on the landscapes, we should focus on management goals that maintain both of those types of reproduction strategies. And one way to do that is to do exactly what Bob Campbell suggested, and is to do big, large treatments and make sure we've got diploids and triploids, sexual and asexual reproducing genotypes in those stands. So before I take any questions, I'd like to make some quick acknowledgments. The T.W. Daniel Endowment, which funded my time while I was doing this work, Richie Gardner, and I need to also thank Bob Campbell for helping me out with this work, and of course my wife and my daughter for helping out with the all-important field work. So with that, I'll take any questions if there's time.
Yeah, there's time for, let's do time for one question.
Go ahead.
What's the hypothesis of the origin of the triploid again? Is it somatic?
No. So Karen can speak to that, but...
It's unreduced gametes. Oh.
What's that?
An unreduced gamete sometime back.
Yeah, and probably a long time ago. Pleistocene?